All right. So experimental design always involves the art of compromise. There's never enough money, never enough time, never enough experimental subjects where you can do a perfect experiment. So for example, I'm doing a food safety experiment. The perfect experiment means I test every head of lettuce for E. coli, every head. But then I have nothing to sell. I'm broke because I tested every head. That you can't do that. Or I want to run an experiment about exploration to other planets to grow some crops. And I'm just curious about it. And the experiment's going to cost me, $30 billion. I'm curious. I don't have $30 billion to satisfy my curiosity. And there are some experiments that people have dreamed up over the years that have never been funded because they're just too expensive. Or eventually people figure out a way to bring the costs down, or it becomes important enough where people are willing to spend the money. So you have to compromise. A business has to compromise even more. How much data, how much confidence do we have to have that A fertilizer is better than B fertilizer? And we need to make a decision. Are we going to go with A or are we going to go with B? And we can't spend all our time and money chasing after the perfect answer. We need a certain level of confidence, and then we just go with it. Until there's more information, more data, we go with our best information. But you should do your own data collection. You've got to compromise. So let's look at some of the terminology here in, is that fuchsia? Hot pink? Defining the experimental design consists of the following steps. First, you have to identify the experimental unit. It could be an individual plant. It could be an individual plot or group of plants. It could be an entire block if it's a huge experiment. But those are the subjects that will be studied. What are you going to do? So if I'm doing, for example, a pruning technique, I could do pruning technique A on a whole row in my vineyard and prune a little differently on row B. So I've got, 200 plants. That's kind of a long row, whatever. I've got 100 plants. Still a pretty long row, 100 times 6, that's 600-foot row. But still, you get the idea. I've got 100 plants pruned one way, 100 plants pruned another way, and I look at gathering data on those to see how they respond over some period of my experiment. Maybe I have one plant. I inoculate one plant with a particular disease and another plant, and it wouldn't just be one plant, but they're in individuals. I'm looking at, let's say, 10 plants, but individually I'm looking at them one at a time, inoculated with a particular disease or not inoculated with that disease, or a close relative of that disease, and I look at what happens to those plants, one plant at a time. So that's my experimental unit. What am I studying? And in agriculture, it's usually a plant, a small group of plants, or a block of plants. And then we have to identify the variables, what are called the constant, the dependent, and the independent variables. We'll get to that in a minute. Then we have to define the treatment structure. So for example, like a fertilizer experiment, maybe I'm going to do fertilizer A versus fertilizer B. Maybe I'm only interested in fertilizer A, but I'm going to do it with 50% fertilizer nitrogen, 50% nitrogen, 75% nitrogen, 100% nitrogen, and 125% nitrogen. So I have 4 levels of nitrogen that I'm going to look at. Those are my treatments. Again, it could be anything. Fertilizer is an easy one to envision. And then I have to design the actual experiment. So you've got 2 groups going into an experiment, the control group and the test group. And a lot of people mess up on experiments by forgetting to do the control group. Oh, I want to try this new fertilizer. So they try the new fertilizer on an entire block. or an entire planting of lettuce or something. You can't tell if your crop looks really good or really bad or just a little better or a little worse. You don't know why. Your gut's going to say it must be the fertilizer because that's all I changed, but you really don't know. Was there something else going on in the weather? Was there something else with that crop of seeds? Was there, who knows? There could be 100 things going on. So the only way you really know is to have controls. And what I like to do with fertilizer experiments is you leave most of the field the way you always do. You don't change a thing. And then in your experimental units, you have fertilizer groups that are unchanged, fertilizer groups that are changed. And then the unchanged is your control, normal fertilizer routines. Or maybe you're doing pesticides. How effective is this pesticide? Well, I can spray certain parts of the field with just water and other parts of the field with the pesticide. Now, with that kind of experiment, you got to be really careful because you need to understand how to design those kinds of experiments, because if I spray parts of the field and don't spray other parts of the field, the way insects or diseases move through a field can be greatly impacted. You have to have large enough areas with random enough opportunity for infection to occur. So dealing with pests and diseases, you need somebody with some experience. How do you create test groups and control groups? So what is the control? I just have been talking about it, but this is an individual or group of individuals in the experiment that's not tested. It doesn't receive the treatment, or it receives, quote, normal treatment in case of fertilizers. Let's say I'm doing a drug test, and so some people get the drug and some people get a sugar pill, and nobody knows. The doctor doesn't know, and the patient doesn't know who's getting what. It's just a number. It's only the researchers in the background that are collecting data. They know that patient number 275 is getting medicine XYZ, and XYZ is the sugar pill. But patient 382 is getting medicine ZYX, and ZYX is the real medicine. And another patient is getting ZZZ, which is a double dose of the medication. So you can look here. Well, I guess we are the control group. So this group right here that's talking, they're not doing anything. You see these two poor mice, they're buzzing like crazy. They must be the ones getting the real medicine, right? Anyway, you're not supposed to know who the control group is, not even the doctor. And just a quick aside here, there is a gold standard in medical research called the double-blind study. And that's when you study where the doctor and the patient both don't know. Only the researcher knows who's getting the control and who's getting the treatment. And the reason is that doctors, even with subtle body language and tone of voice, can influence the patients. And it's been shown many, many, many times over that if the doctor thinks that they know who's getting the real medicine and who isn't, the patients that the doctor thinks is getting the real medicine actually do better than the patients the doctor thinks is getting the placebo or the fake medicine, the control group. And the only difference is the doctor in the doctor's mind. They've tested the doctor. And so subtle body language, facial cues, things the doctor can't even help can influence a patient's ability to feel better, even if everybody's just getting a sugar pill. So anyway, the controls are used for comparison or reference, so you know what normal is. What are you measuring against? Because you never know what changes in the experiment. Even if you think you don't change anything, everything is kept as consistent as possible. Each experiment is unique. So each experiment needs its own set of controls. It allows us to see the independent variable, if the independent, or take 3. It allows us to see how the independent variable changed the results, and it detects the effects of any hidden variables that you might not see. So don't worry about those last 2 statements in terms of the detail, but you really need a control. That's the bottom line. So here's some examples of controls in a variety trial. I don't just count on the field outside the trial. I actually include plots of the field inside of my trial, because there are things going on in my trial that I can't control, like these small plots, like foot traffic and things that are different than way out in the main part of the field. So when I'm running a variety trial in the field, I always include some of the seed from the field in my variety trial. In most of my variety trial is planted by hand. The field is planted by machine planting. That alone can throw off the results. Another example, in an experimental test for viruses, known samples that contain the virus and known samples that do not contain the virus are run. So that might be the upper right-hand corner here. The upper right-hand corner, the extreme first one, is white. It is a known virus-free sample. If that turns blue, you throw the whole thing out. The next 2 are known virus-containing samples. You have 2 of them because things can go wrong with the sensitivity of the test, but I've got 2 of them to say, all right, both of those should detect the virus. So because I've got clear blue, blue in this upper right-hand corner, the first row, I go, that was a good experiment. That's my proof that the bench science, my mixing of chemicals, my adding of virus and other materials, it all worked properly. If I'm in an experiment testing the efficacy of foliar spray, so I'm doing some foliar nutrients or other foliar spray, I would include the control groups that receive just the inert ingredients. Now, I said earlier in this lecture, just water. Technically, it shouldn't be just water. It should be water plus any spreader, sticker chemicals or adjuvants, and inert ingredients from the original chemical. And that can be hard to do sometimes because many manufacturers won't give you a fake gallon of whatever material that they have, whatever herbicide or pesticide or foliar nutrients, with nothing but the inert or inactive ingredients. Sometimes they will, though. If they know you're doing an experiment, they know you're doing research and there's a lot of money on the line for them, they'll often cooperate and give you a bottle that has the active ingredients taken out. If you don't have that, you do your best. You add water, you add spreader sticker material, you add some buffers, you make sure that it is something that is similar to the chemical without inert ingredients. And the manufacturer will help you there. They should. They always have when I've asked, not always on the first request, but you find the right person, and they always give you a recipe that's close enough for the experiment. Human drug study, the placebo, the sugar pill. Now, clearly you wouldn't give somebody colored squishy sugar candies, but it would be a pill that looks exactly like the other pills. It just contains sugar or something else. It's just a pill without any active ingredient. Something inert, tiny amount of sugar like that is like 3 sips of Coke, so it's not going to affect anybody. We talked about the variables, and I said I'll talk about that in a moment. So there are 3 types of variables. You need to know these 3 terms. The independent variable is what is being changed. In other words, the independent variable is the treatment. So if I've got 4 levels of fertilizer, my normal fertilizer, I've got a 75%, 100%, and 125% rates. Those are my 4 levels. So the fertilizer level is my independent variable. It's what I am intentionally changing in the experiment. Okay, it's not the response. So we call it independent because what do we care about? We care about the plant's response to the fertilizer. We care about what happens. So we call this the independent variable. The dependent variable is the response, and the response is dependent on the treatment. It's dependent on the independent variable. What happens? I add more fertilizer, so I've got the different fertilizer levels, and what am I going to get? I'm going to get certain responses. I'm going to get more leaf material with more nitrogen, more greening with more nitrogen. If I go too far, I'm going to have a reduction in fruit quality and other things. I mean, it depends on what it is. There are lots of things that could be the response, but those responses that we're measuring is the dependent variable. And you have to define those before you even start. If I'm doing a lettuce trial, okay, I'm going to measure the head weight for every head of lettuce properly trimmed. I'm going to measure the core height. I'm going to measure the color, etc. Those are 3 dependent variables that I would measure on any lettuce trial. How big is the head, or how much does it weigh in grams? So it's mass technically. I'm going to measure its core height. I'll measure its diameter as well. That's important. And I'm going to measure the, what did I even say? Weight, core height, doesn't matter. I don't know which ones I said, because there are lots of other things. We have rankings for things like ribbiness, and rankings for shape, and rankings for flavor that are based on certain criteria. All of those things on our list to be measured is our dependent variables. What are we going to look at? You can't measure everything, so you look at what things matter in this experiment. And so you have, usually in agriculture, lots of dependent variables. But not too many. You start getting past 8 or 10, you got to ask yourself, why am I having so many? And then there's the constant variables, factors that are kept the same throughout the experiment. It's not the same as a control. So for example, in my fertilizer experiment, I'm going to apply exactly the same amount of water. I'm going to give every plant exactly the same soil type. I'm going to give every plant exactly the same solar radiation. Everything to the best of my ability. I'm going to keep everything else as constant as possible. Those are my constant variables, the things I'm trying not to change. And I don't care how perfect you are as an experimental design person. You can't be perfect in your constants. The soil's going to vary a little bit from here to there to over there. You can't avoid that. The amount of wind and exactly how many insects bit that plant versus the one 2 plants down. You can't make everything perfect and exact. It's just not possible. And so we try to keep things constant, but there's something called experimental error, which is those things we're trying to keep constant, but we can't. And so there's always a little bit of noise in the data coming from our dependent variables. So for example, if I didn't do an experiment at all and I grew with the most uniform lettuce seed I could find on planet Earth, and I grew 100 plants, and I used the same soil in the same greenhouse, only in the middle of the greenhouse, I wasn't near the walls or edges, and I tried to grow every single plant exact. And then I went and I measured head weight, core height, ribbiness, diameter, shape, color. Are they all going to be exactly the same? Every plant? Am I going to get 100 plants that are identical and don't vary at all? Of course not. There's always this intrinsic, unavoidable variation, this noise, no matter how much you try to keep constant variables constant. And that's called experimental error. We'll get to that in a minute. And I'm going to take another break.